Today we will be reading about chromosomal abnormalities, that is variations from the basic genetic complement of chromosomes. These chromosomal aberrations or abnormalities constitute different types. First of all, we have the syndromes. A syndrome is a term which is used for a cluster of phenotypic characters. A cluster of phenotypic characters which are caused due to increase or decrease in the chromosomal number. That is, when we take the autosomes, the autosomes are the somatic complement of any particular cell. If you take a normal human being, we have 24 pairs, 22 chromosomes plus 22 chromosomes is equal to 46 chromosomes. We have 22 pairs of chromosomes and they constitute the autosomes. In addition to the autosomes, we also have the allosomes. The allosomes are otherwise termed as the sex chromosomes, and the sex chromosomes as implicated include the X and the Y chromosomes. So the X and Y chromosomes or the sex chromosomes are called the allosomes. Then we have the autosomes. We are looking into what happens if there is a change in the chromosomal in the chromosomes of human beings. Any change in the chromosomes of human beings can be by increase or decrease of a single chromosome, or increase or decrease of a set of chromosomes. So these sort of variations are normally termed as aneuploidy. Aneuploidy means increase in the chromosomes. The increase in the chromosomes is termed as aneuploidy. So the objective of today's class is, while looking into chromosomal abnormalities, we would be touching upon several issues which are termed as Down syndrome, first is the Down syndrome. Next, we have the Klinefelter syndrome. Later on, we have the Turner syndrome. So we have to make a comparative analysis of Down's, Klinefelter's and Turner syndrome, wherein we exactly know what happens to the chromosome number, what is the meaning of the term non-disjunction. That is, when chromosomes do not separate, when do not segregate or separate during cell division, there is non-disjunction. And due to non-disjunction, there can be an increase or decrease in the chromosome numbers, especially in the syndromes. So the first of the studies deals with chromosomal abnormalities or syndromes, wherein we are comparing the three, the Down's, Klinefelter's and the Turner syndrome. The second component of the class relates to identification of anemic conditions. That is, we will be studying about the genetics of anemia. Later on, we will also be studying the genetics of rhesus factor, that is, how RH is inherited. And later on, we will also be studying about the genetics of color blindness. All these three related topics can be linked to a topic termed as sex linkage. That is, it could be either specific to the male or the female. So, this pattern of inheritance is termed as sex-linked inheritance. So, on one side we have the syndromes, on the other side we have sex-linked inheritance, and anything which is being taught, any sentence which is being taught over here can come in as an objective bit. So, hence a student should very clearly be knowing about what are allosomes and what are autosomes. What do you understand by non-disjunction? What exactly is the differences between the Down syndrome, Klinefelters and the Turner syndrome in terms of both the phenotype as well as the genotype. Lot of questions can come in from the phenotype and the genotype of the three important types of syndromes. And when it comes to color blindness, rhesus factor or anemia, the pattern of inheritance. How is the character transmitted from the parents to the offsprings, and who are the carriers? Of the particular gene of inheritance, and whether the gene of inheritance is dominant or recessive would be studied. So, this forms the basic background or the introduction to chapter of chromosomal abnormalities or syndromes. Now, we are going into the first of the syndromes, which is termed as a Down syndrome. The Down syndrome is otherwise termed as the trisomy of chromosome 21. It is also otherwise termed as a trisomy of chromosome 21. That is, there are 3 copies of chromosome 21 over here. There is the presence of an extra chromosome of chromosome 21. So, chromosome 21 has an extra chromosome. So, an extra chromosome is added. There is an add-on of the chromosome. Extra chromosome is predominantly it is added. Now, normally in any human being we have 46 chromosomes. 2n is equal to 46. So, in Down syndrome it is 2n is equal to 47. 2n is equal to 47. Thereby, we understand that the diploid number 2n plus 1 exists in the case of the Down syndrome. In the case of the Down syndrome, we have other than the diploid number of 46 chromosomes, we have 1 chromosome extra. It is also designated as 2n is equal to 47. Thereby, the offsprings have 47 chromosomes. That is, when a male and a female, when they cross them, you find that always in the F1 the offsprings have 47 chromosomes. The reason we have 47 chromosomes is there is non-disjunction. Or non-separation of the chromosomes occurs. 2 of the chromosomes, they do not segregate clearly. There is non-disjunction or non-separation of any of the 22 chromosomes during the formation of the egg and the sperm cell. During the formation of the egg and the sperm cell, there is non-disjunction or there is non-separation of the chromosomes. Now, when you take Down syndrome, Down syndrome is a congenital disorder. That is, the character can be inherited. The character can be inherited because we have 23 plus 1 chromosome in the egg. Thereby, in the zygote, we would be having 46 plus 1 is equal to 47 chromosomes. So this is a very important feature. They can sometimes be asking you how many sort of where is the extra chromosome coming in? It adds to the 23 pair at the region of the egg. So totally we have got 47 chromosomes. Hence this is also termed as autosomal trisomy. So all the points primarily written on the board indicate objective bits. Now when you take an autosomal, so Down syndrome is an example of autosomal trisomy, and Down syndrome can be exhibited in the form of two important features. It could be 45 plus, it could be 45 plus XY. It is seen in the male, or else it can be 45 plus XX as it is seen in the female. Now this bit is a popular sort of a question which is generally asked in objective bits. When you have got a 45 chromosome plus an XY, it results in the formation of a male, or a 45 plus XX, it results in the formation of a female. Thereby, always in the female, the chromosomes are X, X and X. Thereby, it is termed as a trisomy. In the female, it is X, X and X, and it is termed as a sort of a trisomy. Down syndrome is an example of aneuploidy too. Because a few or extra chromosomes are added. So the chromosomal nomenclature of the Down syndrome, when you take the chromosomal nomenclature of the chromosomal nomenclature, is always 47, or it could also imply a plus 21 situation. So there are two important types of chromosomal nomenclature for Down syndrome. It can be designated as plus 21 chromosomes, or it could be designated as 21. And normally when you take the Down syndrome, the symptoms of the Down syndrome in the form of a cluster of characters are as follows. The phenotype of people with Down syndrome is they show a short stature, again people who inherit Down syndrome have mental retardation, they exhibit mental retardation, they have a round face, and a broad head. Muscles are flaccid. Small ears. They have a protruding tongue. Epicanthal folds are present on the eyes. That is webbing, epicanthal folds occur on the eyes. The entire syndrome is otherwise termed as Mongolian idiocy. This is a popular bit which normally comes in a medical entrance. So, the Mongolian idiocy shows short stature, mental retardation, round face, flaccid muscles, protruding tongue, and epicanthal folds on the eyes. The lifespan of the individual is 8 to 10 years. And the frequency of occurrence of the Down syndrome with the mother is, especially if the mother's age exceeds more than 35 years. So, late marriages wherein the woman attains an age of greater than 35 years, there is a high probability that the child will inherit and it will show a sort of a Down syndrome due to non-disjunction of chromosome number 21. So, these are the specific genetic traits or the human clinical genetic traits of the Down syndrome. After the Down syndrome, we are going into the next syndrome, which is termed as Klinefelter syndrome. We have the Klinefelter syndrome. The second type of a chromosomal abnormality which we are seeing in today's study of clinical or human genetics. Klinefelter syndrome shows a trisomy of XXX type. It's also a trisomy and it shows the feature of XXX type. So, there are 47 chromosomes. There are 47 chromosomes in total. That is, the 47 chromosomes are 44 plus X, X and X. So, it gives rise to 47 chromosomes. So, Klinefelter syndrome is common, especially if the egg has an abnormal XX and it combines with a normal Y chromosome. The second situation could be is if there is an abnormal Y chromosome, when it could be XY. So, a situation of an abnormal XX or an abnormal XY, both of these can lead to increase in chromosomes with XX and X, which forms 47 chromosomes in the case of the Klinefelter syndrome. The basic feature of the Klinefelter syndrome is it results in the formation of sterile males. So, it is predominant in the males. Klinefelter syndrome can easily be identified, especially when you take a scrap of the epithelial cells on the cheek in the interface cells. In the interface cells, they are distinguished due to the presence of a bar body. So, bar body is a very important indicator of Klinefelter syndrome. The clinical symptoms of Klinefelter syndrome exist as the males are normal. They are normal. However, they have a very low sperm count. They are subfertile. That is, they have very small testes. They have developed breasts. Secondary sexual characters are developed in a male, and they exhibit a feminine pitched voice. They show a feminine pitched voice. Limbs are long. They have long limbs, and they also show knock knees. the knees are folded this way. This primarily refers to knock knees. And they have rambling talkative the words which they spell out are in odd. So it is primarily it is a sort of syndrome which shows rambling talkative nature. And the probability of occurrence of a Klinefelter syndrome in a normal population is 1 by 500. So normally the most important concepts are how does it come through with an abnormal XX or an XY? What is the chromosome complement? Where does it affect? It affects the sterile males. And how is it indicated? It is indicated by a bar body. And what is it popularly termed as? It is termed as a sort of a trisomic X, X and X. So these are the basic features of the Klinefelter syndrome. So Klinefelter syndrome in summary is 47, 44 plus XXX with an abnormal XX or an abnormal XY or. And the features are sterile males, subfertile, developed breasts, female pitched voice, 5 long limbs, and again they show rambling talkative nature, and they are identified by the bar body. Next we go into the third type of a syndrome, which is termed as a Turner syndrome. The next genetic abnormality, we have the Turner syndrome. So in the case of the Turner syndrome, it is designated as 45 and also X. The Turner syndrome is a female genetic order, is a female genetic disorder. While in the case of the Klinefelter, it was the male. In the case of the Turner, it's always the sort of female. So that in the Turner syndrome, there is the presence of 44 plus X. 44 plus X, which is equal to 45 instead of 46. Instead of 46. So always this condition is otherwise termed as monosomy. It is otherwise termed as monosomy. So in a typical monosomy condition, we have the 2n minus 1, or it is designated as 2n minus 1. 2n minus 1. The common symptom here is it shows gonadal. It shows gonadal dysgenesis. And the probability of the occurrence of the Turner syndrome is 1 by 2500 in the female births. In the female births, the probability is 1 by 2500. Now, what are the basic features of the Turner syndrome in terms of clinical features? In Turner syndrome, they show retarded sexual development. There is retarded sexual development. The individual is sterile. Individual is sterile. They show a short stature. They show a short stature. A very important diagnostic feature is the webbing of the skin in the neck region. Webbing of the skin in the neck region, and it is followed also by cardiovascular abnormalities. They also show hearing impairment. Hearing impairment and non-functioning gonadal, that is gonadal dysgenesis, primarily indicated by primary amenorrhea. That is, menstrual cycle is absent. So, absence of menstrual cycle, gonadal dysgenesis, webbing of the skin under the neck, as well as cardiovascular abnormalities and retarded sexual development seems to be the sort of a key feature of the Turner syndrome. We had finished off with relation to studying of three important types: the Down syndrome, the Klinefelter syndrome, and the Turner syndrome. Next, we look into sex-linked disorders. That is, we look into terms like what happens to the chromosomes, sex chromosomes, and how do you have gene mutations in sex chromosomes. Whenever we have mutations, gene mutations at the genetic level in the sex chromosomes, it results in the formation of sex-linked disorders, and the first among the sex-linked disorders which we would be primarily be touching is termed as hemophilia. Hemophilia is a sex-linked disorder. And it is primarily caused by gene mutations. Any change in the number, sequence and kind of the nucleotide at the gene level or at the chromosome level is primarily termed as mutations. When you take hemophilia is a disease which normally occurs in the males. It occurs in the male. It is sex-linked. And it is a recessive disorder. A recessive disorder is due to the presence of a gene small h. The gene small h is responsible for hemophilia, and it is sex-linked because it is carried on the X chromosome. It is present on the X chromosome, and normally hemophilia is popularly termed as the bleeder's disease. It is termed as a bleeder's disease, and it has been one of the sort of diseases which had occurred in the royal family. In the royal family of Queen Elizabeth. And the children of the Queen Elizabeth had primarily inherited the pattern of the disease which is termed as hemophilia. So, in hemophilia, the basic features of hemophilia, which is a sex-linked disorder, is there is no blood clotting. Clotting of the blood does not occur. Hemophilia, the sex-linked inherited disorder, is a sort of a disease wherein there is no blood clotting. Or blood clotting is absent. The reason why the blood clotting is absent is due to the absence of anti-hemophilic granules. No hemophilic granules. Or the hemophilic granules are absent. The hemophilic granules are absent. So these granules or globules are popularly termed as factor 8. So factor 8 refers to hemophilic granules or globules, and the absence of this particular factor results in a disease which is termed as hemophilia A. So hemophilia A is one condition where there is absence of hemophilic granules or globules, and in the presence of another factor called factor 9, which is also absent. Factor 9 is otherwise termed as plasma thrombin factor. So the plasma thrombin factor or factor 8, if it is absent, it results in another disease which is termed as hemophilia B. So there are two categories of hemophilia A and B due to the absence of hemophilic globules or plasma thrombin factor or factor 8 or factor 9. Now we are looking at the pattern of inheritance of hemophilia. The pattern, this is the basis for hemophilia, and now we are looking into the genetics, or how does the gene linked sex linked disorders, what happens when it gets transmitted. When you take hemophilia, which can be written as HA or HE, the females are hemophilic when we have an X chromosome which is hemophilic, which is crossed with another X chromosome which is hemophilic, and it is predominantly due to the presence of a recessive gene. Due to the presence of a recessive gene, and is most instances the mothers are the carriers. The mother is the carrier. So thereby transmission of hemophilia is 50% to the sons, 50% to the sons, even if the father is normal. Even if the father is normal, mother is the carrier, and it is inherited by 50% of the sort of the sons. This is the pattern, normal trend of the sex linked disorder related to hemophilia. Next, we are going into another pattern of inheritance which relates to color blindness. Color blindness is a very important disease or a metabolic disorder which is again a sex linked trait. It is a sex linked trait. Color blindness could be acquired, where environment has got a role to play here, or else it could be congenital, transmitted from the parents to the offsprings. And the pattern of color blindness primarily refers to visual, purple deficiency. people cannot distinguish between red and green. They cannot distinguish between red and green. The visual purple deficiency is due to the interference of the retinal rods. Due to the interference of the retinal rods. The retinal rods have, in the case of color blindness, there is the presence of an inverted retina. An inverted retina occurs in the eye. Thereby, it results in the formation of red-green color blindness. It results in the formation of red-green color blindness. Color blindness is common in males or the fathers. So, color blindness is inherited commonly among the males or in the father. Hence, you call it as a sort of a sex-linked pattern of inheritance. For color blindness, again, mothers are the carriers. The mother is the carrier of the colorblind gene. Color blindness is located on the X chromosome. Is located on the X chromosome. Located on the X chromosome. It is due to the presence of a recessive gene. The gene is recessive, location is X chromosome, mother is the carrier, and the trait is linked to the fathers. And the non-distinguishability between red, blue and green is predominantly otherwise also termed as color blindness. Now, the reason why color blindness is very, very important is it is used as a sort of a screening for pilots, for guards who run the railways, as well as for people who work on the electronics. And so, there is sort of a job screening is done for pilots, guards and electronics if they are unable to distinguish between red, blue and green, and in case their visuality is always grey, then they are rejected in the sort of a screening. While looking into the genetics of color blindness, it is important that we keep in mind these specific things. Color blindness is attributed to the father. The mother is the carrier of the gene for color blindness. Thereby, the female daughter is also a carrier of the color blindness disease. The sons would normally be normal. Though phenotypically you may not be able to distinguish between them. So, they are phenotypically similar. If XX designates a mother, and the crossing looks this way, XY is the father, the daughters, that is the X from here as well as the X from here, these are the daughters. The daughter are the carriers for color blindness. They are the carriers for color blindness. And an XY as well as another XY could always result in normal sons. So, daughters are carriers, the mother is a carrier, and the gene for color blindness is inherited from the father. We will be looking at issues with relation to objective bits in color blindness. Normally in color blindness, as you understand, the woman is the carrier, and a woman with XX has got a color blind. She is a carrier for color blind. She could have a normal vision. Now what happens when a woman who's got a normal vision but is a carrier of color blind gene marries a normal male? The normal male is designated as X and Y. Thereby here we understand, in a subsequent sort of a crossing, we have the X chromosome from the male coming over here, and we have the color blind gene X coming over here, and XXC comes on one side, and then we have XY on the other side. Thereby we understand that always from the man, the XY normal is a normal person, whereas XX, the females are female carriers. So a problem could ask you how is the inheritance pattern with relation to color blindness in relation to any particular woman, and what is the situation in the F1, or what is the situation with relation to the sort of a daughters. Now we understand many times in the offsprings we have an XXC, that is a daughter, or the son, which is XY. In another sort of a situation, we understand, supposing a woman is color blind and she marries a normal person who is XY, what could be the offsprings, who could be the carriers, and where is the color blind gene? This is the second set of problems. So when we segregate, X gets segregated over here, the X and the X can combine to form an XX. Similarly, an XC and an X can combine to form an XXC. So this is an example of a color blind carrier daughter. A XXC indicates the daughter, and she is a carrier for color blindness. And similarly, on a combination situation, we have an X and a Y, and similarly we have an XC and a Y. So this is the color blind son. So sons are always XY. They have the color blind gene, but the thing is, the carriers of the daughters are always the sort of a carriers. So many times in an objective bit, they could always be asking you, how does color blind represent? Color blind represents in the form of a heterozygous condition. Heterozygous condition is a characteristic feature of color blindness. We are going into another study which is termed as sickle-shaped anemia. Sickle-shaped anemia is due to a gene mutation. In the autosomes. And sickle-shaped anemia is an abnormal hereditary disorder. It is an abnormal hereditary disorder wherein instead of the standard HbA, hemoglobin A, there is a variation or an abnormal hemoglobin gets formed, which is termed as HbS. HbS is an indication of sickle-shaped anemia, and the normal condition is HbA. So in HbS, what actually happens? We have the hemoglobin, we have the beta chain. On the hemoglobin, we have the beta chain, and in the beta chain, we have the 6th amino acid. In the beta chain on the 6th amino acid, there is a change, that is glutamic acid. Glutamic acid is replaced by valine. That is, glutamic acid is lost and valine is formed. So, the presence of valine is a very important component or a very important event in the formation of HbS. So, hereby in genetics, thymine is substituted by adenine. Thymine is substituted by adenine in the triplet code. So, in the triplet code there is no thymine, whereas adenine is added. Adenine and valine result in the formation of hemoglobin S. So, this autosomal sickle shaped anemia disorder results in the formation of red blood corpuscles which are sickle shaped, and because they are sickle shaped, they cut through the sort of red blood corpuscles and it results in oxygen deficiency. There is an oxygen deficiency, thereby we have clogged blood vessels. There is clogging of the blood vessels, thereby there is lowering of the span of the red blood corpuscles. Lowering of the lifespan of the red blood corpuscles occur. It results in RBC clumping due to lack of oxygen, RBC clumping, which finally results in vascular obstruction. Due to vascular obstruction, there is a sort of most of the metabolism is lowered. So, hemoglobin S, HbS, is recessively inherited, and it is inherited in the homozygous condition. So, the condition of inheritance is recessive and it is in the beta chain with relation to the 6th amino acid. Glutamic acid is replaced by valine. There is a substitution, thymine is lost and adenine is gained, and primarily sickle shaped anemia result in oxygen deficiency. There is clumping of the red blood corpuscles, clogging of the blood vessels, RBC lifespan is reduced, resulting in vascular obstruction, and predominantly this results in lowered metabolism. Next, inheritance we are trying to study is related to rhesus factor. Rhesus is a blood group and it forms a very important component of an antigenic system. It is a foreign protein, and whenever there is the presence of RH, it is represented as RH is represented as an antigen or a foreign body. RH is an antigen, and the presence of the RH antigen is also termed as a RH factor. In human cells, the RH antigen is present on the surface of the red blood corpuscles. It is present on the surface. It is a surface antigen. RH is a surface antigen on the red blood corpuscles. Presence of RH is attributed to being a single gene which is termed as RH plus. RH plus indicates dominance. it is a dominant gene. It is a dominant gene, and Rh minus indicates that it is a recessive gene. It is a recessive gene, and both are in any individual, a man or a woman, due to the presence of an Rh positive or negative. Phenotypically they are just similar. So phenotypically there is no variation. They are phenotypically similar. Individuals with Rh positive or Rh negative are phenotypically similar. We are looking at a situation wherein we are going to study Rh in terms of incompatibility. In terms of incompatibility. Now, Rh incompatibility is a situation which can occur in two situations, which can occur under two conditions. First is during blood transfusion. And second, it can also occur during pregnancy. How does Rh attribute to pregnancy when the first child is born, as well as when the second child is born? So we will have to be studying Rh incompatibility in terms of blood transfusion. Situation, context number 1, context A, and next is the context B, wherein we want to know exactly what happens to Rh during pregnancy and how does Rh affect the sort of being born alive or with anemic conditions of either the first child or the second child. Based on the reason why we are studying all the concepts together is all the concepts are interrelated. So when all the concepts are interrelated, you could have in a multiple choice a cluster of questions which could relate to many of the concepts together. That is primarily the sort of a reason we are trying to cover the concept as a basic unit, and later on it will be followed by a question banks. So we are looking at Rh incompatibility during blood transfusion. During the first blood transfusion, if ever we have an Rh male, and there is a blood transfusion to an Rh minus female, there is no harm. It does not lead to any lethality, because the Rh female develops anti-Rh plus factors, or antibodies in response to the antigen in the blood. So this is termed as the first reaction. It is termed as a first reaction. During the second blood transfusion, that is when the individual gets the transfusion the second time, the antibodies destroy the Rh plus RBCs which enter in. They destroy the RBCs enter in, or they do not allow the Rh plus RBCs of the donors, and thereby it leads to lethality. So during the second blood transfusion, it results in more with relation to lethality. We would now be looking at two situations. What happens with relation to the Rh factor during the first pregnancy, During the first pregnancy, the father's blood has Rh plus. It stimulates the formation of anti-Rh antibodies. Anti-Rh antibodies which are present in the Rh minus or in the mother's blood. Now, these anti-Rh antibodies are not produced in sufficient number. So, they are produced in less in number. They are produced less in number. So, they are not present in enough quantities, and thereby they do not harm the fetus. They do not harm the fetus or the baby because there is no contact. There is no contact, and despite blood transfusion, the baby does not suffer. Baby is safe during the first pregnancy. So, first pregnancy, we have an Rh from the father's blood getting transfused, anti-Rh antibodies are formed in rhesus minus mother's blood. The antibodies are less in number. They are less in number. There is no contact, no harm of the fetus, and the baby is safe. During second pregnancy, Rh plus directly gets into the fetus. The anti-Rh factors are present in the mother's blood. They destroy the RBCs in the fetus, and thereby the child either dies or remains as anemic due to lack of oxygen deficiency. So, here we have an Rh mother, Rh minus mother. We have an Rh minus mother, and we have an Rh plus Foytus reaction. So, primarily during second pregnancy, the reaction is between the Rh minus mother and the Rh Foytus reaction. We understand that in the Rh Foytus, an Rh antigen, in this we have an Rh antigen. We have an Rh antigen on the blood cells, and the maternal antibodies are IgG. IgG are the maternal antibodies. So, the maternal antibodies IgG cross the placenta. They cross the placenta and destroy the Foytus RBCs. They go through the placenta and destroy the RBCs. They destroy the RBCs during the second pregnancy. Thereby, during Rh2 pregnancy, we understand the babies are red blood corpuscles. We have the babies red blood corpuscles and the IgB antibodies, they cross the placenta. It results in the formation of a hemolytic disease. It results in the formation of a hemolytic disease. Hemolytic disease. The hemolytic disease is otherwise termed as erythroblastus foetalis. Is otherwise termed as, the hemophilic disease is otherwise termed as erythroblastus foetalis, wherein the newborn babies can either survive but they tend to be anemic. So this is termed as hemolytic disease of the newborns. Is termed as the hemolytic disease of the newborns. So erythroblastus foetalis hemolytic disease is related primarily with relation to the second pregnancy, when we have the maternal antibodies entering into the placenta and destroying the red blood corpuscles of the baby.